Hello, and welcome back to the For Science Salah Lotfi channel. It's always a pleasure to have you with us as we explore the latest in scientific advancements and celebrate the brilliant minds driving innovation. Today, I want to take a moment to express our sincere gratitude and heartfelt congratulations to the researchers behind an outstanding study titled Enhancement of Fibrinolytic Enzyme Production from Bacillus subtilis via Immobilization Process onto Radiation, Synthesized Starch Dimethylaminoethyl Methacrylate Hydrogel. This groundbreaking research was conducted by a team of highly esteemed scientists. First, we have Professor Dr. Amini Rafat, the corresponding author, from the Polymer Chemistry Department at the National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority, based here in Cairo, Egypt. Joining her in this research is Professor Dr. Eman Arabi from the Microbiology Department, also at the National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority. And finally, we have Professor Dr. Salah Lotfi, also from the Polymer Chemistry Department at the same institution. Their collaborative efforts have led to significant advancements in the production of fibrinolytic enzymes, which are crucial in various medical applications, particularly in the treatment of blood clots. This study is an excellent example of how interdisciplinary research can yield practical benefits for both the scientific community and society as a whole. The article was first received on May 31st, 2011, underwent revision on September 4th, 2011, and was accepted just 5 days later, on September 9th, 2011. It became available online on September 16th, 2011. This research showcases the potential of radiation synthesis and hydrogel technology in enhancing enzyme production, a field that holds promise for future developments in biotechnology and medicine. Once again, we extend our heartfelt thanks and congratulations to Professor Dr. Amini Rafat, Professor Dr. Eman Arabi, and Professor Dr. Salah Lotfi for their outstanding contributions. Your work is truly inspiring and continues to push the boundaries of what is possible in science. Thank you all for tuning in to the For Science Salah Lotfi channel. If you found this video interesting, be sure to like, share, and subscribe to stay updated on the latest scientific breakthroughs. We'll see you in the next episode.
Imagine a world where life-saving enzymes are readily available, produced sustainably, and highly effective. This is the promise held by hydrogels, a fascinating class of materials revolutionizing enzyme production. Like cosmic nurseries where stars are born, hydrogels provide the perfect environment for cultivating beneficial enzymes. These enzymes, the workhorses of the biological world, are crucial for countless processes, from breaking down food to treating diseases. One area where hydrogels are making a significant impact is in the production of fibrinolytic enzymes. These enzymes, capable of dissolving blood clots, are vital for treating conditions like stroke and heart attack. By enhancing the production of these enzymes, hydrogels offer a beacon of hope for millions worldwide. This essay delves into the fascinating world of hydrogels, exploring their potential in revolutionizing enzyme production and transforming healthcare. Join me as we embark on a journey to uncover the power of hydrogels and their role in shaping the future of medicine. The creation of hydrogels is as intriguing as their applications. One method, gamma irradiation, stands out for its elegance and environmental friendliness. Imagine harnessing the power of gamma rays, a high-energy form of electromagnetic radiation, to assemble intricate hydrogel structures. This is precisely what scientists have achieved, using gamma irradiation to initiate the polymerization of monomers, the building blocks of hydrogels. Unlike conventional methods that often rely on harsh chemicals and generate significant waste, gamma irradiation offers a cleaner and more sustainable alternative. It's like using starlight to build a house, leaving a minimal environmental footprint. This eco-friendly approach aligns perfectly with the growing need for sustainable solutions in biotechnology. By embracing such green technologies, we inch closer to a future where scientific progress goes hand in hand with environmental responsibility. In our quest for enhanced enzyme production, we turn to the microscopic world, where tiny organisms hold immense potential. Enter Bacillus subtilis, a remarkable bacterium known for its ability to churn out an array of enzymes, including the coveted fibrinolytic enzymes. These microscopic factories are nature's gift to biotechnology, providing a sustainable and efficient means of enzyme production. Bacillus subtilis, like diligent workers on a production line, can be cultured in large quantities, making them ideal for industrial-scale enzyme production. Their remarkable ability to secrete enzymes directly into the surrounding environment simplifies the extraction process, further enhancing their appeal. These tiny organisms, invisible to the naked eye, hold the key to unlocking a future of abundant and accessible enzyme therapies. Their potential in healthcare, particularly in the realm of blood clot treatment, is nothing short of revolutionary. Now imagine providing these microscopic factories with an ideal living and working environment, a place where they can thrive and maximize their enzyme production. This is where hydrogels come into play. These remarkable materials, composed of networks of cross-linked polymers, act as supportive scaffolds for Bacillus subtilis, providing them with a stable and nurturing environment. Think of hydrogels as microscopic apartment complexes, providing comfortable living spaces for our enzyme-producing bacteria. Within these cozy confines, Bacillus subtilis can multiply and produce enzymes with enhanced efficiency. The porous nature of hydrogels allows for the easy transport of nutrients and oxygen, ensuring the well-being of these microscopic workers. This symbiotic relationship between hydrogels and Bacillus subtilis forms the cornerstone of enhanced enzyme production. Section 5. The Gift of Time, Extended Enzyme Production. One of the most remarkable benefits of immobilizing Bacillus subtilis within hydrogels is the extension of enzyme production time. In traditional fermentation methods, enzyme production is often limited by the lifespan of the bacteria. However, within the protective embrace of hydrogels, Bacillus subtilis can continue producing enzymes for extended periods, significantly increasing overall yield. It's akin to extending the workday for our microscopic enzyme factories, allowing them to produce more life-saving enzymes without the constraints of a limited lifespan. This extended production time translates to greater efficiency and cost-effectiveness, making enzyme therapies more accessible to those in need. The implications for industrial-scale enzyme production are profound, paving the way for a new era of abundance. Section 6. Enhanced Stability. Conquering temperature and pH challenges. Enzymes, while incredibly powerful, are also delicate molecules, sensitive to changes in temperature and pH. This sensitivity often limits their applications, particularly in industrial settings where harsh conditions are commonplace. However, immobilizing enzymes within hydrogels provides a solution, enhancing their stability and expanding their range of applications. Think of the hydrogel as a protective shield, safeguarding the enzymes from the harsh realities of the outside world. This shielding effect helps maintain the enzyme's structural integrity, allowing them to function optimally, even under challenging conditions. This enhanced stability is crucial for industrial applications where enzymes are exposed to extreme temperatures, pH levels, and other denaturing agents. By overcoming these limitations, hydrogels unlock the full potential of enzymes in various industries. Section 7. Reusability, the industrial advantage of immobilized cells. In the realm of industrial enzyme production, reusability is key. Traditional methods often involve using enzymes only once, leading to high costs and significant waste. However, immobilizing Bacillus subtilis within hydrogels introduces the concept of reusability, transforming the economics of enzyme production. Imagine our microscopic enzyme factories, safely housed within their hydrogel apartments, being reused multiple times without compromising their productivity. This is the power of immobilization, allowing for the recovery and reuse of Bacillus subtilis, significantly reducing costs and waste. This sustainable approach aligns perfectly with the principles of a circular economy, minimizing environmental impact while maximizing resource utilization. This paradigm shift in enzyme production opens doors to a future of sustainable and affordable biotechnologies. Section 8. Fibrinolytic therapy, dissolving blood clots and saving lives. Now let's shift our focus to a specific application where hydrogel-enhanced enzyme production holds immense promise: fibrinolytic therapy. Blood clots, while essential for wound healing, can be life-threatening when they obstruct blood flow in vital organs. Fibrinolytic therapy, using enzymes to dissolve these dangerous clots, has emerged as a cornerstone of treatment for conditions like stroke and heart attack. Imagine a treatment that can swiftly and effectively dissolve blood clots, restoring blood flow and saving lives. This is the power of fibrinolytic therapy, and hydrogels are playing a crucial role in enhancing its effectiveness. By boosting the production of fibrinolytic enzymes, hydrogels are paving the way for more effective and accessible treatments for these life-threatening conditions. The potential to improve patient outcomes and save lives is immense. Section 9. Microbial enzymes. Tiny workers with huge therapeutic potential. Beyond fibrinolytic therapy, the potential of microbial enzymes in medicine extends far and wide. From treating digestive disorders to developing novel cancer therapies, these tiny workers are proving to be invaluable allies in our fight against disease. Hydrogels, by enhancing the production and stability of these enzymes, are unlocking their full therapeutic potential. Imagine a future where microbial enzymes produced sustainably and abundantly using hydrogels form the basis of numerous life-saving therapies. This is the vision driving research in this field, a vision where the power of nature's tiniest workers is harnessed to address some of humanity's greatest health challenges. The possibilities are vast and inspiring. Section 10. A future brimming with possibilities. Hydrogels in therapy and industry. As we delve deeper into the world of hydrogels and their role in enzyme production, we begin to grasp the sheer scope of their potential. From revolutionizing healthcare to transforming industrial processes, hydrogels are poised to leave an indelible mark on our world. Imagine a future where sustainable, hydrogel-based enzyme production platforms become commonplace, providing a constant supply of affordable and effective therapies. This is not merely a distant dream, but a tangible reality within our reach. The convergence of material science, biotechnology, and nanotechnology is propelling us towards a future where the lines between biology and technology become increasingly blurred. This future, driven by innovation and a deep understanding of nature's intricate processes, is brimming with possibilities. Section 11. Conclusion: a universe of potential within a tiny gel. In the vast expanse of scientific exploration, few discoveries hold the transformative power of hydrogels. These unassuming materials, composed of simple building blocks, have opened up a universe of potential in enzyme production, with far-reaching implications for medicine, industry, and beyond. Like stars illuminating the cosmos, hydrogels are shedding light on new frontiers in biotechnology, guiding us towards a future where life-saving therapies are readily available, industrial processes are sustainable, and the boundaries of scientific innovation are constantly pushed. As we continue to explore the depths of this fascinating field, one thing remains certain: the future of hydrogels, like the universe itself, is filled with wonder and endless possibilities. And within these tiny gels lies the potential to solve some of humanity's greatest challenges. Hey there, science fans! We explored a groundbreaking study on hydrogel and enzyme production. Now we're going behind the scenes to uncover the how, the materials, and methods that make this research possible. Think of a scientist like a master builder. Before constructing a skyscraper, you need a detailed blueprint, right? That's exactly what the materials and methods section of a scientific study is: the blueprint for discovery. It outlines every ingredient and every step taken, ensuring that the experiment can be replicated and the results can be trusted. Without clearly defined materials and methods, scientific studies would be like a cake recipe missing key ingredients and baking instructions. It'd be chaos in the kitchen. So, buckle up as we break down the precise steps and ingredients that make this hydrogel and enzyme research so exciting. In this scientific adventure, our star players are two incredible materials: soluble starch and DMAEMA, short for Dimethylaminoethyl Methacrylate. Don't let the long name scare you. DMAEMA is just a type of molecule that loves to link up with others. Think of starch as long chains of sugar molecules, kind of like edible beads on a string. It's a natural polymer found in plants like corn and potatoes. We use it every day in foods like bread and pasta. DMAEMA, on the other hand, is synthetic, made by scientists in a lab. These two materials are the key ingredients in creating our hydrogel, a superabsorbent material. We'll get into how they work together in a bit. Remember, just like a delicious cake needs flour and sugar, our experiment needs starch and DMAEMA. Now let's meet the tiny but mighty hero of our experiment. This microscopic organism is a type of bacteria found naturally in soil and the human gut. Don't worry, this little guy is a friendly bacterium. It's like a microscopic factory, capable of producing amazing enzymes. Enzymes are like tiny machines that speed up chemical reactions. They're essential for all sorts of processes, from digesting food to breaking down pollutants. In our experiment, we're particularly interested in the enzymes produced by that can break down starch. To study these enzymes, we need to provide our tiny factories with the perfect environment to thrive. This involves carefully cultivating and maintaining them in a nutrient-rich broth, ensuring they are happy and productive. Remember those building blocks, starch and DMAEMA? It's time to put them to work in creating our hydrogels. This process is a bit like making jelly, but with a scientific twist. First, we dissolve our starch and DMAEMA in water. Then we use a cool process called gamma irradiation. Imagine tiny, invisible rays of energy, like miniature lightning bolts, zapping through our mixture. This irradiation helps link the starch and DMAEMA molecules together, forming a network. This network acts like a sponge, able to hold a lot of water. And voila, we have our hydrogel. The exact ratio of starch to DMAEMA, as well as the duration of irradiation, are carefully controlled to create hydrogels with specific properties. Now that we have our hydrogels, how do we know how well they turned out? That's where the gel fraction calculation comes in. It's a way to measure how much of our initial starch and DMAEMA actually formed into the hydrogel network. Imagine taking our hydrogel and squeezing out all the water. What's left is the solid gel part. By comparing the weight of this dried gel to the initial weight of the starch and DMAEMA we used, we can calculate the gel fraction. A higher gel fraction means a more successful hydrogel formation. It tells us that most of our ingredients were effectively incorporated into the network. This information is crucial for optimizing the hydrogel preparation process and ensuring consistent results. Hydrogels are amazing because of their ability to absorb and hold water, just like a sponge. But not all hydrogels are created equal. Swelling studies help us understand how much water different hydrogels can absorb and how fast they do it. In these studies, we take our hydrogels and immerse them in water. Over time, we carefully measure how much weight they gain as they soak up the water. This gives us valuable information about their swelling capacity. The swelling behavior of a hydrogel depends on its composition, the ratio of starch to DMAEMA used. By tweaking this ratio, we can fine-tune the hydrogel's properties for specific applications, like controlled drug delivery or tissue engineering. Remember our microscopic factories, It's time to put them to work. But first, we need to prepare them for their enzyme-producing mission. This involves creating an inoculum. An inoculum is like a starter culture, similar to what you'd use to make yogurt or sourdough bread. We take a small amount of our carefully maintained culture and add it to a fresh nutrient broth. This allows the bacteria to multiply and thrive. Once we have a healthy and active inoculum, we introduce it to our hydrogels. Here's where things get really interesting. They actually become immobilized within the hydrogel network. It's like they set up shop within the sponge-like structure. Now that are happily settled in their hydrogel homes, they can start producing those valuable starch-degrading enzymes. But how do we actually measure how active these enzymes are? That's where human fibrin comes in. Fibrin is a protein found in our blood that's responsible for clotting. It's what helps stop bleeding when we get a cut. In our experiment, we use fibrin as a substrate, a material that our enzymes can act upon. We mix our enzyme-loaded hydrogels with a fibrin solution and measure how much fibrin is broken down over time. The more fibrin that's degraded, the more active our enzymes are. This assay provides a quantitative way to assess the effectiveness of our enzyme production system. Science is all about finding the best way to do something. In our quest to maximize enzyme production, we use a technique called repeated batch fermentation. It's like running multiple rounds of our experiment, tweaking the conditions each time to see what works best. Imagine our enzyme-producing setup like a tiny brewery. We start with a batch of our hydrogel containing the immobilized. This is our fermentation vessel. We provide the bacteria with all the nutrients they need and allow them to produce enzymes. After a set period, we harvest the enzyme-rich liquid surrounding the hydrogel. This is our precious product. We then replenish the nutrients in the fermentation vessel and start the process again. By repeating this process multiple times, we can optimize factors like nutrient concentration and fermentation time to achieve the highest possible enzyme yield. Just like Goldilocks and the Three Bears, enzymes have their preferences. Temperature and pH play a crucial role in how well enzymes function. Too hot, and they can break down. Too acidic or basic, and they might not work efficiently. In our experiment, we carefully control both temperature and pH to find the sweet spot where our starch-degrading enzymes are most active. We do this by conducting enzyme activity assays. Remember the fibrin test? At different temperatures and pH levels. By plotting the results on a graph, we can identify the optimal conditions for enzyme production. This information is invaluable for scaling up our process and ensuring that our enzymes are always working at their best. We've talked a lot about the structure of our hydrogels, but how do we actually see something so small? That's where the amazing technology of scanning electron microscopy, or SEM, comes in. It's like a super-powered microscope that uses electrons instead of light to create images. Imagine shrinking down to the size of an ant and exploring the surface of our hydrogel. That's what SEM allows us to do. It provides incredibly detailed images of the hydrogel's porous structure, revealing the intricate network of starch and DMAEMA. These images help us understand how the hydrogel's structure relates to its properties, like its swelling capacity and its ability to immobilize. It's like getting a sneak peek inside the microscopic world of our experiment. And there you have it, science fans. We've journeyed through the fascinating world of hydrogel and enzyme production, exploring the meticulous materials and methods that make this research possible. From the careful selection of starch and DMAEMA to the optimization of enzyme activity, every step plays a vital role in achieving meaningful results. Just like a strong foundation is essential for building a skyscraper, rigorous methodologies are the bedrock of scientific discovery. By clearly outlining every material and every step, scientists ensure that their experiments are replicable, reliable, and contribute to the ever-growing body of scientific knowledge. So, the next time you hear about a groundbreaking scientific breakthrough, remember the importance of the materials and methods behind it. It's the unsung hero of scientific progress, paving the way for a brighter future. Hydrogels are fascinating materials. They consist of a network of polymers. These polymers can absorb and retain significant amounts of water. This gives hydrogels a soft, jelly-like consistency. This unique characteristic makes them very attractive for various biomedical applications. Hydrogels can mimic the properties of natural tissues. This makes them suitable for drug delivery, wound healing, and tissue engineering. Hydrogels can be made from natural or synthetic polymers. They can be designed to respond to specific stimuli. This includes changes in temperature, pH, or the presence of specific molecules. This responsiveness makes them ideal candidates for controlled drug release systems. They can deliver medication directly to the target site. Among the diverse range of hydrogels, DMAEMA hydrogels stand out. DMAEMA stands for Dimethylaminoethyl Methacrylate. It is a monomer that can polymerize to form hydrogels. DMAEMA hydrogels possess unique properties. These properties make them particularly suitable for biomedical applications. DMAEMA hydrogels are pH responsive. This means their swelling behavior changes with the pH of the surrounding environment. At low pH, the DMAEMA groups become protonated. This leads to increased swelling of the hydrogel. This pH sensitivity is particularly useful for drug delivery applications. It allows for controlled release of drugs in specific environments. Radiation-induced cross-linking offers a versatile method to synthesize DMAEMA hydrogels. This innovative approach is gaining traction in various scientific fields due to its efficiency and effectiveness. This technique utilizes high-energy radiation, typically gamma rays or electron beams, to initiate the cross-linking process. These high-energy sources are capable of penetrating deep into the material, ensuring uniform cross-linking throughout the hydrogel. The radiation generates free radicals within the DMAEMA monomers. These free radicals are highly reactive species that play a crucial role in the cross-linking process. These free radicals then react with each other, forming cross-links between the polymer chains. This results in a three-dimensional network that imparts unique properties to the hydrogel. This method offers several advantages over conventional chemical cross-linking methods. For instance, it eliminates the need for additional chemical agents, which can sometimes introduce impurities or unwanted side reactions. It is a clean and efficient process that does not require the use of toxic cross-linking agents. This makes the entire procedure safer for both the environment and the researchers involved. This makes it particularly suitable for biomedical applications where purity is paramount. Hydrogels produced through radiation cross-linking are often used in drug delivery systems, wound dressings, and tissue engineering. Furthermore, the degree of cross-linking can be precisely controlled by adjusting the radiation dose. This level of control allows scientists to fine-tune the mechanical and chemical properties of the hydrogel. This allows for the tailoring of the hydrogel's properties to meet specific application requirements. Whether it's for increased elasticity, enhanced biocompatibility, or specific degradation rates, radiation cross-linking provides the flexibility needed for advanced material design. Gel fraction and swelling behavior are two critical parameters that dictate the performance of DMAEMA hydrogels. These parameters are essential for understanding how these hydrogels can be used in various applications, from medical to industrial. Gel fraction refers to the percentage of polymer that has been successfully cross-linked into the gel network. This cross-linking process is crucial because it determines the mechanical strength and stability of the hydrogel. A higher gel fraction indicates a more robust and stable gel structure. This means the hydrogel can maintain its form and function under various conditions, making it suitable for demanding applications. Swelling behavior, on the other hand, describes the hydrogel's capacity to absorb and retain water. This property is particularly important for applications where the hydrogel needs to interact with biological tissues or fluids. This property is influenced by various factors, including the degree of cross-linking, which can be adjusted to control the swelling behavior. The hydrophilicity of the polymer, which determines how well the hydrogel can attract and hold water molecules, and the pH and ionic strength of the surrounding medium. These environmental factors can significantly affect the swelling capacity and overall performance of the hydrogel. Swelling measurements provide valuable insights into the hydrogel's network structure and its potential applications. By understanding these measurements, scientists can tailor hydrogels for specific uses. Hydrogels with high swelling ratios are often desirable for drug delivery and tissue engineering applications. These hydrogels can expand significantly, allowing them to hold and release therapeutic agents over time. They can accommodate large amounts of therapeutic agents or cells within their porous structure. This makes them ideal for delivering drugs or supporting cell growth in tissue engineering, providing a versatile platform for medical advancements. A home for cells immobilization within hydrogels. Cell immobilization is a crucial aspect of various biotechnological and biomedical applications. It involves trapping cells within a matrix. This allows for their use in bioreactors, biosensors, and tissue engineering constructs. DMAEMA hydrogels, with their biocompatible nature and tunable properties, serve as excellent matrices for cell immobilization. The hydrogel's porous structure provides a three-dimensional environment. This mimics the natural extracellular matrix. This environment allows cells to attach, grow, and function effectively. Moreover, the hydrogel's ability to encapsulate cells while allowing for the diffusion of nutrients and waste products makes it an ideal platform for creating artificial tissues and organs. Hydrogel porosity impact on enzyme production. The porosity of DMAEMA hydrogels plays a crucial role in enzyme production. Enzymes are biological catalysts. They accelerate biochemical reactions. Immobilizing enzymes within hydrogels offers several advantages. This includes enhanced stability, ease of separation from the reaction mixture, and reusability. The hydrogel's porous structure provides a large surface area. This allows for a high density of enzyme immobilization. The pore size is crucial. It needs to be large enough to allow for the diffusion of substrates and products. However, it should also be small enough to prevent enzyme leakage. Temperature and pH guardians of enzyme stability. Enzymes, like most biological molecules, are sensitive to temperature and pH changes. These factors can affect the enzyme's three-dimensional structure, which can lead to a loss of activity. DMAEMA hydrogels can provide a protective environment for immobilized enzymes, helping to maintain their stability over a wider range of temperatures and pH values. The hydrogel matrix can act as a buffer, resisting changes in pH. This is particularly important for enzymes that are sensitive to pH fluctuations. Furthermore, the hydrogel network can restrict the enzyme's movement, reducing the likelihood of denaturation at high temperatures. The gift that keeps on giving: reusability of DMAEMA hydrogels. One of the significant advantages of using DMAEMA hydrogels for enzyme immobilization is their reusability. Traditional enzyme immobilization techniques often result in a loss of enzyme activity after a single use. However, DMAEMA hydrogels can be used multiple times without significant loss of enzyme activity. This is because the hydrogel matrix protects the enzyme from harsh conditions and prevents its loss during the reaction process. The hydrogel can be easily recovered from the reaction mixture by simple filtration or centrifugation. The recovered hydrogel, with its immobilized enzyme, can then be reused in subsequent reactions. This reusability significantly reduces costs and enhances the efficiency of biocatalytic processes. Future horizons: exploring new applications. The unique properties of DMAEMA hydrogels make them promising candidates for a wide range of future applications. These hydrogels are synthesized through a meticulous process that ensures their high quality and functionality. Their biocompatibility and pH-responsive behavior open up exciting possibilities in drug delivery. This means that they can interact harmoniously with biological tissues and respond to changes in pH levels, which is crucial for medical applications. They can be used to develop smart drug delivery systems that release drugs in response to specific stimuli, allowing for targeted drug delivery to specific tissues or organs. This targeted approach minimizes side effects and maximizes therapeutic efficacy, making treatments more effective and safer for patients. Furthermore, their ability to support cell growth and function makes them attractive for tissue engineering applications. These hydrogels provide a conducive environment for cells to thrive, which is essential for creating functional tissues. They can be used to create scaffolds for growing artificial tissues and organs, which holds immense potential for regenerative medicine. This could revolutionize the field by providing solutions for organ shortages and improving the quality of life for patients needing transplants. Researchers are exploring the use of DMAEMA hydrogels for various other applications, including wound healing, biosensors, and gene delivery. These hydrogels can accelerate the healing process, provide real-time health monitoring, and facilitate the delivery of genetic material for therapeutic purposes, showcasing their versatility and potential in advancing medical science. Conclusion: DMAEMA hydrogels, a boon for biomedicine. DMAEMA hydrogels represent a versatile class of materials that hold immense potential for various biomedical applications. Their biocompatibility, pH responsiveness, and ability to support cell growth make them ideal candidates for drug delivery, tissue engineering, and enzyme immobilization. As research continues to unravel the full potential of DMAEMA hydrogels, we can expect to see even more innovative applications of these remarkable materials in the field of biomedicine. They offer a promising avenue for developing new and improved therapies that will improve human health and well-being.
Hello everyone, and welcome back to the For Science Salah Lotfi channel. Today, we want to take a moment to express our heartfelt thanks to all of you, our dedicated viewers, for your continuous support and engagement with our content. Your passion for science is what drives us to keep exploring and sharing the latest in scientific research. We also want to extend our deepest gratitude to the brilliant researchers behind the study we're highlighting today. The research is titled, "This study represents a significant step forward in the field of polymer chemistry and microbiology." Allow me to introduce the remarkable team behind this research. Professor Dr. Amani Rafat, who is the corresponding author, from the Polymer Chemistry Department at the National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority, located in Cairo, Egypt. Professor Dr. Iman Arabi, from the Microbiology Department at the same esteemed institution. And of course, our very own Professor Dr. Salah Lotfi, also from the Polymer Chemistry Department at the National Center for Radiation Research and Technology. Their collaboration and dedication have led to this groundbreaking study, which explores innovative methods to enhance enzyme production, a crucial aspect in various biotechnological applications. This significant research was published after a rigorous review process. Here's a brief overview of the publication details. Article history: received May 31st, 2011, revised September 4th, 2011, accepted September 9th, 2011, available online September 16th, 2011. Keywords: cell immobilization, fibrinolytic enzyme, starch, dimethylaminoethyl methacrylate, hydrogel, radiation. Their work was published in a reputable scientific journal, contributing valuable knowledge to the scientific community. Once again, thank you to Professor Dr. Amani Raifat, Professor Dr. Iman Arabi, and Professor Dr. Salah Lotfi for their exceptional contributions to science. And to all our viewers, thank you for tuning in. Your support is invaluable, and we look forward to sharing more exciting discoveries with you in the future. Stay curious, and see you in the next video.